Obviously, yesterday in lab, I pointed out that we're starting the nervous system stuff, and I want to introduce the nervous system and talk about what the nervous system is in sort of broad terms. We're going to look at a few different ways to divide the nervous system up into more manageable parts. I think it's important at this stage for introducing stuff to make a point about the types of divisions we're talking about here. Some sources are just going to say, oh, there's this and that nervous system, and this and that nervous system, and this and that nervous system. And sometimes try to shoehorn things in together that don't really fit. The divisions we're going to look at are initially anatomical divisions, and then we're going to also consider functional divisions within that. So the first and most obvious division, anatomically speaking, is the central nervous system versus the peripheral nervous system. The central nervous system is simply the brain and the spinal cord, what's contained within the cranium and the vertebral cavity. And then peripheral is everything else. Now, the line between central and peripheral is sometimes a little bit quirky, but for the broad strokes that we're going to consider, it's pretty much simply just the brain and spinal cord versus everything else. And everything else is essentially the nerves that extend away from the brain and spinal cord to go out into the peripheral structures of the body. And then structures that are contain neurons, which I mentioned last time, called ganglia. And with one of the models in lab, I showed you a chain of ganglia that's found alongside the vertebral column. And I also talked about and showed you micrographs of a ganglion that would be at the root of the nerve that attaches to the spinal cord. So that's the broad anatomical definition, central versus peripheral. Now, continuing with sort of the anatomical definition of this, when we're talking about the structures of the nervous system or nervous tissue, we can usually think of them as divided into two major groups. There are bundles of fibers, the axons from neurons, that come from neurons and travel from one place to another. And then the groups of the neurons, the cell bodies themselves. When we're looking at the central nervous system, we call a bundle of fibers a tract. And when we look at, we call a group of neurons a nucleus. In the periphery, those structures that I just mentioned that basically make up the periphery are really just that, bundles of fibers, which we call nerves, and groups of neurons, which we call ganglia. And ganglia is plural, ganglion is the singular of that. And we'll just find those structures throughout the periphery. Another way to think about those structures, which is more relevant to the central nervous system than the peripheral nervous system, is using terms gray matter and white matter. I'm actually going to go to a picture in a little while to distinguish those things, but it's also really an anatomical depiction of the stuff within the nervous system. Sorry, I got left out of the series of slides. But then there's two ways of thinking about the functional divisions in the nervous system. And these are not mutually exclusive. It's just two different ways of thinking about the nervous system, separating it into different functions. the first one is sort of about the broad functions of what the system does, the somatic nervous system versus the autonomic nervous system. Somatic, which comes from the word that means body, is referring to the voluntary and conscious aspects of what our nervous system does, which I mean by that, I mean our conscious awareness of what's around us and our voluntary control of our skeletal muscles primarily. And then the autonomic nervous system is really involved in homeostasis. It's the involuntary aspects of how we control what's going on in our body. It's largely subconscious, unconscious, whatever you want to, however you want to describe it. You're not aware of a lot of the processes going on there. And then it's responsible for controlling everything but skeletal muscle. So that's going to be cardiac muscle and smooth muscle, as well as glandular tissue. So if you want to run, that's a conscious decision, and you're going to control skeletal muscles that'll make your legs move and you'll run. But as you're running, you're going to be making demands on tissues in your body, so the autonomic system will go along with that, speed up your heart rate, increase your breathing, deliver more oxygen to those skeletal muscles. You'll start to sweat to help keep yourself cool while you're exerting yourself and all those sorts of things. So functionally, there's two different groups of, or I shouldn't say groups, there are two different systems involved in what's controlling things, but they do often work together. Another functional definition of the nervous system is to describe it as having sensory and motor components. So sensory is going to be referring to what comes into the system, and motor is describing the output of the system. The word motor usually refers to movement, controlling muscles, but it does sometimes get included in a more broad idea of response. So sweating is a motor response. There's no muscles that's causing you to sweat, but controlling those glands is, could be considered a motor response. In that functional definition of sensory versus motor, there are some sort of gray areas between it. Think about something complex like driving your car. The sensory processes involved in that are going to be your visual sense of what's occurring in the world around you, your auditory sense, listening for things like sirens or whatever that might be coming from further away than you can see. You're going to have the tactile sensation of your hands on the steering wheel and your feet on the pedals, and the sensation of moving those body parts to manipulate the car. All of that sensory input. The motor output is the actual moving things and causing the car to drive around. But there's actually a bit in between there that can't strictly be called sensory or motor. For example, if you hear a siren, you're going to probably respond to that by looking around and trying to find the flashing lights. But you're not necessarily going to see them right away, so you also are going to process the sound. You have a way of trying to localize where sound comes from. You can hear when things come from one side of your head versus the other, so you can try to figure out where things are. While it's sensory input that feeds into those things, it's a little bit more complex what's happening in figuring out where the sounds are coming from and tying the sound to a visual cue like flashing lights or something like that. And then also using that information to decide what to do. So if there's an emergency vehicle coming up behind you, flashing its lights and its sirens going, then you should be looking to pull over to a safe spot so that they can drive by and impede it. All of that to get to the motor response of moving the car out of the way, figuring out where the things are coming from, deciding what you need to do in response to that, isn't purely sensory or purely motor. So there is sort of another aspect within the functional definition there that's sometimes referred to as integrative or, integrative. For a second I was thinking there's another word that I used there, but I think in the book, integration is the term for that. So those are two ways of looking functionally at what's going on, and they're not mutually exclusive. There are sensory components of the somatic nervous system, and again, those are your conscious perceptions for the most part. So I don't really have to describe that. Things that you are aware of happening in your environment, those are somatic senses. But there's also autonomic senses, and this is a good example of what I meant by the autonomic system being largely subconscious or unconscious, however you want to describe it. One of the things the autonomic system regulates is blood pressure. You don't have any conscious perception of your blood pressure. You don't feel hypertensive or hypotensive on any given day. But you have sensors in your bloodstream, in specific blood vessels, that respond to increased pressure causing the walls of the blood vessels to stretch out, and the motor output from that is going to speed up or slow down the heartbeat so there's less pressure going through the system. You're not aware of that, but there is a sensory input, the stretch receptors in blood vessels, that cause the motor output to change cardiac muscle activity. And then motor, like I said, is usually thought of in terms of muscle control, but there are some things that are responses that aren't technically muscles. Obviously, if it's skeletal muscle control, we would consider that somatic nervous system motor function. And if it's cardiac or smooth muscle or glandular tissue, then we'd consider that autonomic nervous system responses. So these overlap quite a bit. Now, this picture is trying to put a lot of these ideas together in terms of looking at the anatomy that I sort of started off with, the picture with the brain, the spinal cord, and nerves and ganglia. Just to show you that there is, in the central nervous system, somatic and autonomic processing of sensory stimuli. So that's senses and the somatic autonomic functions. So all of that maps together. You can look at a certain place in the brain that's responsible for processing somatic sensory stimuli. So that's a very directed way of putting all of these things together. Or you can look at a specific place that looks at autonomic sensory processing. You can look at that in the periphery. There are ganglia in certain places that receive sensory information and relay that onto different parts of the nervous system, et cetera, et cetera. And the same can be said about motor responses being directed by different parts of the nervous system. I add one thing in here just to mention it, which is kind of an additional anatomical definition or distinction within the nervous system. It's called the enteric nervous system. It's really sort of a subset within the periphery. It is the nerves and ganglia that are associated with your digestive tract. Enteric means guts, basically, what's inside of you. And so this is the nervous system that's responsible for controlling your digestive function. It's actually a very extensive network of nervous tissue. I don't recall exactly where I found this, and I can't verify it, but I seem to remember at some point in my career coming across some mention that there's as much nervous tissue in the enteric nervous system as there is in the brain or central nervous system. I don't remember which it is, but there's quite a bit of nervous tissue there. It's just integrated into your stomach and intestines and all that stuff. So it's not as obvious as the organ that's your brain, but it's still going to contain a lot of nervous tissue. So it's a big part, anatomically speaking. It's got its own sort of properties to it because it can sort of work on its own to regulate digestive function, but it really is an extension of the peripheral nervous system, or I should say a specific region of the peripheral nervous system. It's an extension of part of the autonomic nervous system for the most part, because again, it's not conscious perceptions, and it's controlling things other than skeletal muscle, mainly smooth muscle and glands that are in the digestive tract. So it fits in there, but it is a special kind of part that I want to point out here. And I've added to the picture a little stretch of small intestine just to represent part of the digestive tract there. Now I want to take a second out to come back to the terms I introduced a little while ago, which were tract, nucleus, nerve, and ganglion. So I need to add into the presentation stuff here just a simple 4-square table where we have groups of neurons or bundles of fibers in either the central nervous system or the peripheral nervous system. So what do we call a group of neurons in the central nervous system? What did I just tell you? Yeah, that's what we call a nucleus. And what's a bundle of fibers in the central nervous system? Tract. What's a group of neurons in the peripheral nervous system?
Ganglion.
Okay. Singular is ganglion. And what's a bundle of fibers in the peripheral nervous system?
The nerves.
So this is an easy 4-square table just to lay out those particular definitions. The picture I have up on the screen right here is getting at one issue I want to make here, which is the word nucleus. In this course, we use the word nucleus in 3 different contexts. At the beginning of the semester, we started talking about chemistry, and so we referred to where there are protons and atoms. And that's the nucleus of the atom. It's the center of the atom. And then when we started talking about cells, we start to think about the nucleus of a cell, which is sort of the center of the cell. In this particular picture, it's not the exact middle center of the cell, but it is the controlling center of the cell. It contains the DNA and directs a lot of what the cell does. Now we're using the word nucleus to refer to part of the brain in this picture, central nervous system in general. And so this red outline is highlighting a particular nucleus in the brain. Now this is an MRI image, and with MRI you can distinguish between gray matter and white matter. They appear differently on the MRI. So the gray parts that we see here, that's gray matter, and there's one region of gray matter that's outlined right there, which is a nucleus. And then you can see, for instance, white matter that's sort of deep inside the brain. I'll have a picture to show you gray and white matter again in a while. But, gray matter is essentially the nuclei in the central nervous system. We don't always call them nuclei. For example, there's gray matter on the outside edge of the brain, kind of covering the outside, a wrinkly part of the brain, like we see here. We wouldn't call that a nucleus because it's organized in its own sort of way, but it's related to what a nucleus is. The particular nucleus that's highlighted here, I chose just because it's a nice single example of a nucleus, but it happens to be the nucleus that's often related with reward systems. You may have heard reference to the pleasure center of the brain or something like that. So that nucleus is often active related to when we're internally rewarding ourselves for doing something. So you finish your homework on time, or you go for a jog, or whatever it is, those kinds of things will activate areas in there. They release neurons in there release chemicals called endorphins. Endorphin means internal morphine. So morphine is an addictive drug, and it was discovered that it works because there are chemicals inside of our brain that cause the same changes, and they're released by this area of the brain as sort of a reward. The reward center that we're looking at here, interestingly, is often associated with addiction. So morphine or something like that's an addictive drug because it stimulates this part of the brain. There's also a lot of neurons that are related to the use of a chemical called dopamine, which is very important in addictive drug behavior. It's directly tied to cocaine and related drugs activity in the brain, and that's the area, the nucleus, that does it. Now, I'm sort of describing what this nucleus does because in all of these cases, the word nucleus sort of means the center of something. Here it's the center of the atom. Here it's the center, if not physical, at least functionally, center of the cell. And here we're talking about a group of neurons that are a central location for a particular function. So this particular nucleus is sometimes referred to as the reward center of the brain. And that's why we use the word nucleus. Now, I'm pointing this out because it is sort of your responsibility as a student to pay attention to the context in which the word nucleus is used so that you know how to apply it. I have certainly asked questions of students at various times that have to do with the word nucleus, and they don't appropriately figure out the context of the use of that word. And so I might ask something about, what do you find in the nucleus of a cell? And the answer I get is protons. You find protons in the nucleus of an atom. You find DNA in the nucleus of a cell. Now, you could potentially make the argument, well, there's protons in DNA, so obviously it's in. That's not the kind of thinking you need to put into figuring out how these work. Consider the context of how a word like this is used and answer the questions that might be associated with it in keeping with that context. Okay, so a nucleus that contains protons is an atomic nucleus. A nucleus that contains DNA is a cell nucleus. A nucleus that contains neurons is a nucleus in the central nervous system. Okay. Now, I also want to talk about the terms nerve and tract. They're both describing what we call a bundle of fibers, axons that are all bundled together that travel from the source where some neurons are located to the target. When that bundle of fibers is in the peripheral nervous system, we call it a nerve, and when it's in the central nervous system, we call it a tract. But we could actually be talking about the same particular structure, and this is largely a geographical distinction. So in this picture, I'm showing you the pathways for the visual system. So we have here the eyes with these dots are representing where neurons in the retina are located. And then the lines extending away from them are the axons that are going into the brain to deliver that sensory information to the brain. The bundle of fibers that extends from the eye towards the brain is referred to as the optic nerve, using the term for a peripheral bundle of fibers. And then once they cross this line right here, which is anatomically called the optic chiasm or optic chiasma, where these fibers cross over from one side to the other, any bundle of fibers we see posterior to that point in this picture is called the optic tract. It's one axon or one bundle of axons, okay, going through the optic nerve and continuing on into the optic tract, or in the optic nerve here and crossing over and continuing the optic tract. It's just that there's this magic line where the optic chiasm is, that's the boundary between peripheral and central for the visual system. That would not apply to other systems. This is just for that particular system. It's sort of like roads having different names depending on what town you're in. Okay. Route 5 comes up through the Pioneer Valley and crosses through a lot of the major cities, towns, whatever you want to call them in the area. What do you call Route 5 in West Springfield? Riverdale. Okay. What do you call it in Holyoke? Does anybody know? Is that too far north for people?
I'm sure.
It's called Northampton
Northampton.
Road because it's going to Northampton. But when you get into Northampton, if anybody goes that far north, what do you call it? As soon as you cross the line from Holyoke to Northampton, it's called Pearl Street. And then actually in Northampton, where Route 5 crosses Main Street, south of Main Street, it's Pearl Street. North of Main Street, Route 5 is now called King Street. Same road, same asphalt, if you will. It's just a matter of geography, where you are, what it's called. Same thing here. The axons that are going from the retina back into the brain, immediately when they leave the eye, we call them the optic nerve. Same axons, nothing's changed. They cross this magic line of the optic chiasm, and now we call them part of the optic tract. Nothing's changed. They're still the same fibers. It's just a matter of where we're looking at them, what we call them. So I pointed out something special about the word nucleus, and I pointed out an understanding about tract and nerve. The fourth term here is ganglion. There's nothing special to point out about that. It's just the term for groups of neurons found in the peripheral nervous system. For some animals, usually invertebrates like worms and bugs and crustaceans and those sorts of things, they don't have a very advanced nervous system. So instead of having a brain, they just have a primary ganglion, just to suggest that it's not as complex as our central nervous system. And sometimes all it is they have nerves running the length of their body, and every so often there's a little cluster of cells in a particular place. That's a ganglion. We still use that term describing peripheral structures for us, but we have a much more complex central nervous system, so we use nucleus and tract to refer to that. Oh, there's those. Okay. So yesterday in lab, I introduced what nervous tissue is, and I just want to talk about some of that in a little bit more detail. So nervous tissue, like I defined last time, is made up of neurons and glia. And, oops. There we go. I was trying to stop it from cutting off the terms there. Neurons are the cells that we usually associate with the electrical activity that's so important for communication in the nervous system. And then glia, like I described them yesterday, were originally thought of as just supporting cells. And the word glia from Greek really means glue, in the sense that glia just seem to hold things together, to be the sticky parts. I talked last time, I want to reiterate it again here, that glia will probably be discovered to be much more important, even in electrical communication in the cells, in the tissue. But at this stage of understanding, for the sake of a textbook presentation, we're going to stick to glia as being supporting cells. But the nature of this class is going to have you probably looking up things on the internet. So understand that there's more out there about glia than we're going to discuss in this class. And be wary of that. So if you're going to be looking things up about glia to answer questions in this class, be especially careful that you make sure that it fits within the context of this course. Okay? It is true that in the last few or several years, there's been some serious data suggesting that glia are also involved in electrical signaling in the brain nervous system. However, we're not going to concentrate on that. We're going to stick to their supportive role because that data is not very well established, and we don't really understand that aspect of glia enough to cover in a introductory course like this. So be careful of what you find online that is describing glia. You might very well be able to find multiple sources that say the same thing independently, satisfying our common knowledge definition that I introduced at the beginning of the semester. But here I want to caution you just to be especially aware of what fits into the course versus what's sort of on the cutting edge of information that we know. And if you're ever in a situation where you're not quite sure if you understand what you're reading, if it's going beyond the context of the course, please contact me and I'll give you a little bit of I'll give you feedback on how to understand what you might be coming across, which might come down to don't bother looking at that, look somewhere else, or I might be able to explain a little bit more in depth what a source you found is describing. So, and we looked at this picture last time, just lays out the structure of the neuron, the dendrites surrounding the cell body, and the single axon emerging from that cell body. Neurons are polar in the sense that they receive at one end and they send information out at the other end. And introducing that idea of polarity, that the neurons have a directional aspect to them, we can think of this cartoon representation that I showed you last time of unipolar versus bipolar versus multipolar cells. So again, unipolar cells, information flows from dendrites in one location through the axon that travels directly to the target. And it kind of bypasses the cell body itself because the axon is a bifurcation of the one process coming off of the unipolar cell. Now I want to say a little bit more about unipolar cells than I did yesterday, which is we don't actually have true unipolar cells in our body. This is not actually a true unipolar cell. This is more appropriately called a pseudo-unipolar cell. And I want to mention that because you might see that in material you come across. So a true unipolar cell is a cell body with no dendrites and an axon coming off of it. So, vertebrate animals do not have true unipolar cells. We find these sorts of things in invertebrates. Probably the most famous example is in this mollusk known as a sea, it's either a sea cucumber or a sea slug. Technically, I guess it could be either. And all it is that synapses come in and connect directly to the cell body from other sources, and then the axon sends the information out. That's truly a unipolar neuron. As a cell body, there's only one thing coming off of it. The pseudo-unipolar cells that we're looking at still have the same basic structure. It's just instead of this being the axon that goes off, it splits into two pieces. One end has dendrites, and the other end goes to the target to make a synapse. Okay. And the unipolar neurons that we're looking at here are pretty much always the same kind of neuron. They're sensory neurons. Their dendrites are in the location where sensory stimuli show up, and their targets are usually somewhere in the central nervous system. So for the sense of touch, the dendrites are going to be in the dermis of the skin, and the axon will end in the spinal cord of the brain where it's relaying that sensory information. The cell bodies of these unipolar neurons are always located in ganglia outside the central nervous system. Most of them are going to be in the dorsal root ganglia, which I mentioned last time. Although there are a few ganglia associated directly with the brain, which we'll learn about what those are later in the course. But they're another alternative place where unipolar neurons would be found. Not all senses use unipolar neurons. Two senses that we're really going to talk about more in AMP2 are where we find bipolar neurons. One of those is the sense of smell, the olfactory sense. And its dendrites are in the nasal mucosa and interact directly with odor molecules. And then the axon goes up into the brain, carrying information about smell there. Another one is found in the retina. There's a cell that's literally called the bipolar cell, and it's found in the retina, and it connects photoreceptors to the neurons that carry information from the retina into the brain. And again, that's something we're going to talk about next semester as we get into sensory systems. The multipolar neurons come in a bunch of different shapes and sizes. We looked at several of them last time. The motor neurons of the spinal cord were the first ones that we looked at, which are the motor unit neurons, the ones that control muscle contraction directly. We looked at the Purkinje cells, which are big neurons in the cerebellum that are important for how the cerebellum does its thing. And we're going to talk about what the cerebellum does later on in, I think, the next chapter or the chapter after that. So we'll look at what Purkinje cells actually do for the cerebellum there. In looking at the cerebellum, I also showed you the granular cells from the granular layer. And we couldn't see any detail in them because they're such small cells, but they're in fact also multipolar cells. They have a bunch of little dendrites sticking out of the cell body and then one axon that usually goes and synapses on the dendrites of the Purkinje cell. And then I showed you pyramidal cells. We didn't look at that under the microscope, but I showed you some pictures from a Google search of what they look like. And they're often found in the outer covering of the cerebrum, the cerebral cortex. But those are all different versions of multipolar neurons. Okay. And the cartoon image that we have here is usually representative of one of those motor neurons. And then here's just some more specific pictures. I showed these last time, the pyramidal cell, the Purkinje cell. We have the bipolar cells of the olfactory system at the bottom here. And then these are central neurons of the olfactory system that are receiving that stimuli information. But they are, in fact, multipolar cells, a different type. They have 4 things coming off of them, which classifies them as multipolar. But they're unlike the other types that we've seen here. Then we want to look at, think about glia. Now, I quickly sort of showed you this list last time, and I want to spend a little bit more time with it today. There are 6 different types of glial cells that we're going to consider in this course, and 4 of them are found in the central nervous system, meaning the brain or spinal cord, and 2 of them are found in the peripheral nervous system, meaning outside. I've lined them up in these 2 lists, and there's a table just like this in the book, in a specific way, because astrocytes in the central nervous system and satellite cells in the peripheral nervous system are similar types of cells. Now, the similarity is actually fairly superficial, in that they're the cells that support neurons primarily. Astrocytes do a lot of things that satellite cells don't have to do or need to do because they're in the periphery. Astrocytes we usually associate with regulating the ion concentrations in the extracellular fluid, as well as helping process neurotransmitters and clearing things out of synapses and those sorts of things to aid with the electrochemical signaling that we'll be talking about later. Also, very importantly, astrocytes are involved in what's called the blood-brain barrier. For the central nervous system, there's a barrier that limits what can move from the blood into the brain directly. We call that a privileged blood supply. Interestingly, the only other significant privileged blood supply that I know of is the testes blood barrier. So that might tell you something about the relationship between brains and testicles. Nobody? Come on. Anyways, a privileged blood supply is in place because you want to limit the possibility of possible toxins or pathogens getting into particular locations. for the brain, we want to limit the chance of central nervous system infections or toxicity, certainly, because that's going to mess up how neurons do their job, which can mess up very fundamental things about us, our memory, our personality, our ability to generate motor responses at a very basic level. A lot of things that we want to avoid, so there's a privileged blood supply there. Since I brought up the testicular the blood-testes barrier, the point there is that sperm production happens sort of continuously, and any toxic or pathogenic element that's introduced might create mutations in the sperm DNA, which will propagate as mutations in offspring. So that needs a very tight barrier. The relationship between those barriers is actually just in the privileged blood supply. It has nothing to do with any function. I just like that joke, but apparently none of you did. So, but astrocytes are very important in the blood-brain barrier, mostly because they associate directly with the blood vessels going through the brain and are part of regulating what can move out of the blood into the brain. Essentially, all that can move out of the blood into the brain are water molecules, charged particles like ions that are going to be very important, carbohydrates, mainly glucose, and amino acids. Pretty much, and lipids. Pretty much everything else is kept out of your brain. An important aspect of the blood-brain barrier comes down to drug design. If you're going to create a drug that's going to have an effect on the central nervous system, you have to create it to be able to pass through the blood-brain barrier. A common drug to treat Parkinson's disease is called L-DOPA, which is an amino acid that can cross the blood-brain barrier. There's an amino acid called tyrosine, which is converted into L-DOPA in your body. And then L-DOPA is used to make dopamine, which is very important in the functions that are lost with Parkinson's disease. You cannot just give somebody dopamine and expect that to get in the brain. It will not cross the blood-brain barrier because it's not an amino acid. It's derived from an amino acid, but it's not an amino acid itself. So to be able to give somebody a drug that's going to increase dopamine levels in the brain and recover the function that's lost in Parkinson's disease, you have to give them L-DOPA because being an amino acid, it can cross the blood-brain barrier. And then once you get L-DOPA levels high enough in the brain, it'll be made into dopamine where it's needed to counteract the effects of Parkinson's disease. So astrocytes are very important in establishing the blood-brain barrier. The next pairing we have here, oligodendrocytes and Schwann cells, I put them side by side because those are the cells that insulate the axons. They provide the myelin that wraps around the axon itself. Really what myelin is it's cell membranes of these cells wrapping around the cell over and over again. We'll see some pictures of that later on. In the peripheral nervous system, that's done by Schwann cells. In the central nervous system, that's done by oligodendrocytes. So they do the same sort of thing. How they do it is slightly different. And I'll talk about the differences when we look at those in more detail. But they do the same functional thing. Then microglia and ependymal cells in the central nervous system don't have glial counterparts in the periphery because they are present in the central nervous system because of the blood-brain barrier. And there's not a blood-brain barrier in the periphery, so there doesn't need to be corresponding glia there to play the same role. Microglia are essentially like white blood cells that help fight off infection. There's some evidence that actually they are white blood cells that migrated into the central nervous system during embryonic development, and then they become a special type of glial cell. I'm not quite sure about how that happens or what data really supports that, but I've heard that and it makes sense. So in the mature brain, which means anything postnatal, the blood-brain barrier established by the astrocytes won't let white blood cells cross over in the brain. So you have microglia to help fight off infection in the brain itself. In the periphery, you have white blood cells that are part of the cardiovascular system, and they do that job. Ependymal cells, I showed you an example of those in the microscope slides yesterday. They line the inside surface of the hollow core to the spinal cord and some spaces within the structure of the brain itself. They are kind of like an epithelium, that they're lining the inside of this space, but they're not an epithelium. They are glial cells, nervous tissue, but they play a role in helping to secrete and absorb things across the boundaries of the brain. One place where the blood-brain barrier breaks down is actually where there are some ependymal cells. They make the fluid that we find inside the brain called cerebrospinal fluid because they allow more things to filter out of the brain, out of the blood and into the brain. But they still control things, and you can't, a lot of those toxic or pathogenic things aren't going to be able to get through them. But it's a little leakier than where the astrocytes are established in the blood-brain barrier. But again, nothing like that's necessary in the periphery because the periphery doesn't have that barrier. So components of the blood will actually be able to get directly to the peripheral nervous tissue structures. So we don't need any counterpart in the periphery for ependymal cells. This figure kind of shows what the various central nervous system glia look like. This is an astrocyte. It is named because it looks like a star-shaped cell. Now, personally, I can't say that it looks more like a star-shaped cell than a lot of other cells in the body, but that's where the name came from. And some microscopists looking through the microscope saw that, and however he's named it, said, oh, that looks like a star, I'll call it an astrocyte. That's where the name came from. This is an oligodendrocyte, or a couple of them here labeled. They are responsible for insulating or myelinating axons that are passing through the central nervous system. Oligodendrocyte literally means few branch cell. So it's a cell with a few branches coming off of it. The dendrites that we see associated with neurons, those are these branching things that come off the cell body. In a similar way, functionally nothing like the dendrites on a neuron, but structurally very similar, there are these processes that come off of the oligodendrocyte. They're not called dendrites, they're just called oligodendrocyte processes. They reach out from the cell body, and each one wraps around a little bit of an axon. And here we see both of the examples that are labeled. They have 4 different processes reaching out, wrapping around 4 different spots, 2 on one axon here, 2 from this cell on this axon, and 2 from this cell on this axon. So they kind of all contribute to the myelin sheaths that are found around axons. And many of the myelin sheaths that are drawn in here, like these 2, are not drawn to be connected to a particular oligodendrocyte. And that's just economy of the picture. If they drew in all of the oligodendrocytes that provided all of the myelin sheaths we see in here, then the picture would get even busier than it is. We have a microglial cell up here, and they're called microglia because they're very small for glia, but actually compared to white blood cells, they're probably similar in size to those. White blood cells out in the periphery are usually 12 to 20 micrometers in diameter. Neurons can be 100 or more micrometers in diameter. So a microglia next to a neuron looks very small, but compared to actual white blood cells in the periphery, they're probably a comparable size. And then finally down here in the bottom, we have ependymal cells. The corner of this picture, it's not labeled as such, but would suggest that this would be an empty space within the brain. And so there's a layer of these cells that line that empty space. But then, unlike an epithelium, these cells have processes that reach out and interact with astrocytes and other structures in the nervous tissue. So they're, again, not exactly epithelial cells, they're nervous tissue cells, but they behave or at least structurally, are very similar to those types of cells. This is the closest that we can come up with for a picture that represents the same thing in the periphery. Okay. The picture we just saw is just representative of a sort of simplified slice through the brain, for instance. This is just showing us a unipolar cell, which would be in the ganglia in the periphery. Its axon coming off, which then is going to make up a nerve in the periphery. It's associated with two types of glial cells. The ones that surround the cell body itself are called the satellite cells, and then the ones that wrap around the axon are called Schwann cells. I showed you satellite cells surrounding a unipolar cell body in the microscope slide yesterday. And so you can kind of imagine where the slice is taken through the cell body. You can see the nuclei sort of scattered around like they're orbiting the big cell body. That's where the name satellite cell comes from. Schwann cells wrap around axons, much like I just showed you with oligodendrocytes, except for the fact that the entire Schwann cell wraps around the axon. So there are 4 Schwann cells there at the bottom wrapped around that axon. And I'm actually not sure if the axon extending away from the cell body is wrapped by a Schwann cell or a satellite cell, and it doesn't really matter. But because the insulation, the myelin that's important, is along the 4 that are at the bottom. Those are each individual myelin sheaths that are each made up of one entire Schwann cell. So the cell body is not off to the side somewhere. We can actually see the nucleus of the Schwann cell as part of the myelin sheath there, each of these, and it just wraps around all of that stuff. Question?
How can you tell between the Schwann cells and oligo?
Again, it's geography. You can just call those oligos. It's an easy way to refer to them. If it's in the central nervous system, it's an oligodendrocyte. If it's in the periphery, it's a Schwann cell. Okay.
Is it similar?
Sorry?
Is it similar to each other?
In how we look at tissue, neither one is a very detailed looking cell, so we can't tell too much about them. You will see the nuclei of Schwann cells right adjacent to where the axons are. And you'll see the nuclei of oligodendrocytes technically kind of floating around between the axons. But those two statements are really not that different. Right next to the axon or kind of next to the axon. And we don't see enough detail for either the cells to see their cytoplasm or the cell membrane structure too much. But it's really about, if you're in the brain and the spinal cord, it's an oligodendrocyte. If you're outside the brain and the spinal cord, it's a Schwann cell. There actually is one weird little exception to that. The optic nerve that I showed you earlier, that's the fibers going to the retina, from the retina to the brain. It's called the optic nerve when it's peripheral to the optic chiasm, but it actually has oligodendrocytes in it. The reason for that has to do with embryonic development, and we're going to talk about embryonic development of the brain in the next chapter, so I'll come back to that idea because it is a little quirk there. But otherwise, if it's in the periphery, it's a Schwann cell. If it's in the central nervous system, it's an oligodendrocyte. And that's just sort of the law. You can trust that with the one exception of the optic nerve, which I'll address later on. Here we show sort of how myelination takes place, how these cells wrap around the axons. So at the top we see a Schwann cell, and we can see its nucleus here, that has sort of a groove in it that an axon fits into. During development, what happens is the Schwann cell kind of wraps around and encloses all of the axon in lots and lots of layers of its own cell membrane. And so here at the end, we see that wrapping around and around. There's really not any cytoplasm to speak of in between those layers of cell membrane. So here, we can see the cytoplasm around the whole, within the whole Schwann cell. And in the middle picture, where we start to see some wrapping, we can see that there's cytoplasm within the cell as it's wrapping around. But once it finally wraps around and completely surrounds that axon, the cytoplasm in there is basically going to be squeezed out and just be layers of cell membrane next to each other over and over again. Down at the bottom, what we're looking at here is an electron micrograph of the cross-section of an axon. Now, in electron micrographs, you use a stain which I described for you yesterday that stains lipids. That's the osmium-based osmic acid stain that I mentioned. Wherever we see the black, that's a stain in here that it associates with the lipids. Now, there's actually two stains that you use in electron micrographs. One of them is the osmium stain that shows us where lipids is. You also use a lead-based stain. I think it's lead citrate, but I can't remember. It's been decades since I did this. And the lead stain associates with proteins. Both of these, the osmium or the lead stains, essentially are just heavy metals that block the electrons in the microscope. Electron microscopes work on similar principles to light microscopes, but instead of photons, particles of light, electrons are what are shot through the tissue. And then taking a picture of a light microscope, then the light falls on a, what do we call those things? I can't think of it now, but in a digital camera, there's a little chip that the light falls on. It's how the camera on your phone works. So if you have a digital scope like we have in the lab, there's a camera that the light travels through the specimen towards, and where it hits that. If the lights pass through a stained area, then the wavelength of the light will be adjusted, or not adjusted, but the same wavelength light will be reflected off of that, and we'll see the different colors. In an electron micrograph, what's happening is you're shooting electrons through this, which are much smaller sized particles than photons. And so we can get much higher resolution to the pictures. And wherever we have a big atomic nucleus for osmium or lead, it'll block the electron. So the electron will hit that nucleus and bounce off in a weird direction and never hit the camera chip. So anywhere where we see black here is where an electron was bounced off of one of these heavy metal stains. We know, looking at this, what's lipids and what's proteins because we can see the fine details of the structure. And so this thick black outline here are the layers and layers of cell membrane wrapping around the axon. And then in the axon, you can see it's mostly light. There's not a whole lot there that picks up stain, but there are some little dark spots here and there, which are going to be cytoskeletal proteins running the length of the axon, giving it its structure. And this big kind of clear space right here, we can see a dark outline to it. This is probably a vesicle of some sort that's being transported down the length of the axon. And so it contains something that doesn't pick up any stain and looks empty. But we can see the lipid boundary of the vesicle there with that osmium stain. And then just next to that, this sort of grayish looking thing, and if you look closely, you'll see that there's a little bit of a layered pattern within that. And there's another one up here. Those are mitochondria. Okay, mitochondria on an electron micrograph are very easy to pick out because they have two layers of membrane, one on the outside, sort of defining its boundary, and then on the inside it has a layer of membrane that's kind of all folded up inside. So they're very obvious to see. Those are definitely mitochondria. And then beyond the myelin that we see, any thin black lines we see, which are similar to the thin black line that's the wall of this vesicle, those are single layers of phospholipid bilayer membranes. This is probably part of the Schwann cell membrane. And we can kind of follow it around. I don't know why there's a little bump down here at the bottom. But we can follow it around, and it continues here, This big thing here is probably the Schwann cell nucleus, although it seems really big for what I would expect. And I'm not entirely sure what this picture is of specifically, but we can start to see these kinds of structures. And knowing what we're looking at, we can get a really good idea of that structure because we know what proteins and mitochondria and vesicles and cell membranes look like. We can pick that out pretty well. If you look closely, especially if you're not looking at it on this projected image, but rather directly on the computer screen, you should be able to notice that there's a pattern of layers within the within the myelin here. Because there's layers and layers of cell membrane wrapped around it. And we can see that in the structure of the myelin. Again, it's a little hard to see in the projected image because of how the projector's camera cuts down on some of the resolution. But if you look at it directly on the screen, and this picture is out of the textbook, and I think that the textbook has a link to the source, which is probably that website I shared with you back when we were looking at tissue from the University of Michigan. I think it's a link there. It's possible if the link's in the book, it might be broken because the University of Michigan updated that, but go back to the tissue section in Blackboard, and I have a functional link back to the tissues website at the University of Michigan. Look up myelin or axons or something like that, and you should be able to see something like this there. What I just zoomed to and zoomed back from is moving on to the next aspect of what this chapter gets into, which is looking at the functional idea of what nervous tissue is for. And this is a really good stopping point. I want to sort of today has been talking about the anatomy of nervous tissue, really. Next time what I want to do is I want to move on and think about how nervous tissue functions, which is the electrochemical signaling that I've been talking about here. So let's call it a day here and pick up with this next time, because this is just a very natural place to stop. I could go on from here and spend 10 minutes talking about that stuff, but then I'd have to spend that same amount of time at the beginning of the next class just to remind you what I said. So we'll just save it all for next time and pick up with that to put all of the functional aspect together in one place. So that's all I have for today. And again, we'll pick up with stuff